Assalamualaikum warahmatullahi wabarakatuh. Hi, my name is Azlan. Today I'm going to present our research entitled Do Undergraduate USIM Students with Adequate Oral Literacy Have Better Oral Status? Oral literacy can be simply defined as how an individual make appropriate oral health decision when they read, see, listen, and translate it to their understanding on basic information. Study has shown an association between oral literacy and oral status. People with low oral literacy is at risk of having dental caries and periodontal disease. When we come across with such information related to oral health, do we really understand about the message and making a correct decision? Because misinterpretation of health messages from any medium such as audiovisual, pamphlets or poster, or even online through websites or YouTube can cause harmful effect to one's oral cavity. This is very crucial situation. Thus, it is very important an individual to seek appropriate information of related oral health knowledge from genuine resources, including from dentists. Studies indicated that health students compared to non-health students practice good oral health behavior and having good oral health literacy. This has been confirmed that both variables related to each other. Therefore, it is important for individuals to obtain correct health information to increase their literacy and oral health status. Oral status can be determined as having oral problems such as dental caries and periodontal disease. Low oral literacy is associated with presence of caries and periodontal disease. As we have known about the association between oral literacy and oral status, what about USIM students from various faculty? Do they have better oral literacy? Is their oral status better? Is there any relationship between the variables among USIM students? Here, we are going to answer those related questions. Thus, our objective is to assess the oral literacy level and oral health status among university students. Our null hypothesis is that there is no difference of oral health literacy and oral status among USIM undergrad students. Method and methods. This is a cross-section study conducted from October 2019 to January 2020 among various undergraduate students batch 2019-2020. We occupied a stratified random sampling techniques across different fields of study to get a proper number of subjects. We obtained ethical approval from USIM Ethical Committee for conducting the research. All procedures in this study was consented before data collection. A self-administered validated oral health literacy index questionnaire of Malay version or OHLIM translated by Ramli et al. Was distributed via Google Form before call for clinical examination. We examined for caries and periodontal conditions. Analysis was performed using SPSS. This slide describes about the OHLIM questionnaire used to indicate oral health literacy. The questions consist of reading, comprehension, and numeracy section. It comprised of questions on dental caries and periodontal disease in reading comprehension. For numeracy section, it comprised of drug labeling, dental appointment, and post-extractions of tooth instruction questions. Final scoring of OHLIM then classified as inadequate, marginal, and adequate literacy respectively. For clinical examinations, dental caries and periodontal conditions were recorded using DMFT, which is decade, missing and field index, and modified community periodontal index. The MFT index describes the number of teeth affected, only scored for 20 teeth out of 32 teeth. Wisdom teeth is excluded. The periodontal pocketing is scored as no pocketing, pocketing between 4 to 5 mm, and pocketing more than 6 mm. Periodontal disease in this study was described as healthy or having periodontal pocketing more than 4 mm. Results and discussion. The caries prevalence of USIM students were 58.9% with the median score MFT of 1. Although the prevalence is high, but the median of 1 shows that the caries among the students can be considered as low. Whereas, the prevalence of periodontal disease was 13.2%. The result of caries and periodontal disease showed that students in USIM had practiced good oral hygiene behavior and have fair oral health status. Figure 1 depicts the level of oral health literacy in the different fields of study in USIM. About three-quarters of the students had adequate oral health literacy. Health science and science and technology students had better or adequate oral health literacy than social science students. There was an association between the field of study and oral health status. This finding is similar to what have been reported elsewhere. The reason is because of the exposure to health-related knowledge among health students increased their oral health literacy. For science and technology, there are studies saying that science syllabus had also include health-related knowledge in their study among the students. Across group, caries was reported to be high, whereas parental problem is low, as shown in the figure. We found no association of oral health literacy with dental caries and parental disease, although studies found otherwise. Further study is recommended to obtain concrete evidence. From the result found, we would like to recommend a further study includes more wide subjects among other universities in Malaysia. And to conclude other important variables such as diet intake and smoking status to see the effects on oral health literacy amongst university students. Thus, we would like to conclude that USIM students had adequate oral health literacy. Those with inadequate oral health literacy had a high percentage of dental caries. Thus, it is essential to ensure that the health message delivered is well understood. Improving oral literacy among undergraduate students may help to reduce oral health problems and improve adherence to good oral health behavior. This is our references. With that, thank you.
Assalamualaikum warahmatullahi wabarakatuh. I am Muhammad Haris bin Hasmi. Today I would like to present to you my research on title Study on Smoking and Myocardial Infarction in Malaysia, and this research was done with my supervisors, which is Dr. Yumsuhailah binti Yusuf and Dr. Norhana binti Abdul Rahim. This is the abstract of my study. First of all, the introduction. Ischemic heart disease, or as known as IHD, is the number one cause of death worldwide and in Malaysia. It is the number one cause of cardiovascular disease. Myocardial infarction (MI) is the symptom of IHD. 3.8 million men and 3.4 million women died due to IHD yearly. IHD is the condition when there is a development of waxy substance or plaque inside the coronary arteries. This condition will result in difficulty to supply the heart tissue with blood. Myocardial infarction will occur if this condition reduces and blocks the blood supplies to the heart tissue. In Malaysia, number of CVD cases in National Heart Institute increased by 5% every year. There were 10,000 cases of cardiology and hypertension and 4,000 cases of heart surgery in 1 year. Smoking is the significant risk factors of IHD. 43% of men in Malaysia were smokers and 59% of men aged 21 to 30 years old are smokers. Smoking was the most preventable cause of death worldwide, and it has caused 5 million premature deaths globally each year. The problem arises from this research. First, MI is the main cause of death in Malaysia. The number of people who died due to MI had increased by 54% from 2007 to 2017. Higher rate of tobacco consumption in Malaysia. 5 million smokers in Malaysia were classified as children or younger than 18 years old. More than 50% of male students in secondary school were smokers. A study by Institute for Public Health 2015 had found that more than 30% of smokers smoke more than 25 cigarettes per day. Smokers were likely to be found from the lower income group in Malaysia. B40 households spend 2.3% of their monthly income for tobacco and alcoholic beverage, higher than T20 household, around RM70 per month spent for tobacco individually. The participant of lower income household in family takaful or life insurance is lower compared to higher income household. The reason of people taking insurance or takaful because they want to cover the financial need if any unexpected loss happens in the future. Insurance or takaful act as financial security if such loss occur. The objective of this research is to investigate the association of smoking with myocardial infarction. The methodology. This research used narrative literature review to review papers related to smoking and myocardial infarction. MI definition. According to Tygerson and Searle, there are 5 types of MI, which is spontaneous MI, MI secondary to an ischemic imbalance, MI resulting in death when biomarkers are unavailable, MI related to the precocious to the PCI, MI related to stent thrombosis, and MI related to CABG. And this is the description of the type of the MI. You can read it. Next. Myocardial infarction mortality rate. As we can see from the graph, world 15.8% of deaths in the world was due to IHD, and as we can see, Philippines has the highest rate of prevalence of IHD, which is 18.2%. IHD is the most common cause of death around the world. There are around 3.8 million men and 3.4 million women around the world have died caused by IHD. MI death trend in Malaysia. AHD is the number one cause of death in every state in Malaysia except for Sabah and Perlis. Melaka record the highest percentage of RHD with 19.3%, while Johor and Putrajaya record the highest increase in the percentage of IHD death by 3.5% and 3.1% each. As you can see from the trend of CVD death in Malaysia, there is an increase every year. And the percentage of IHD for every state in Malaysia is higher than 5% for each state. And the 5 principal causes of death in Malaysia is ischemic heart disease, pneumonia, cerebrovascular disease, transport accident, chronic lower respiratory disease. Based on the gender, 60.68% of IHD cases are from male, and based from the ethnic group, Bumiputra had there is 62% of IHD case from Bumiputra, and the prevalence of IHD from urban area is 66%. The principal cause of death by age groups, the IHD is the principal cause of death from the age 41 and above. Risk factors associated with the development of MI. As we can see from the graph, from the table, all of the research mentioned that smoking is the significant risk factors of MI. By analyzing, by controlling the smoking habits or reducing the smoking, the number of cigarettes per day, we can reduce the risk of MI, the risk of having MI, or the risk of death due to MI. The risk factors trend in Malaysia. In Malaysia, diabetes, high cholesterol, and hypertension are the main risk factors of CVD. Smoking and body mass index level also have the significant effects on MI. There are 1.7 billion Malaysians have 3 major CVD risk factors, while 3.4 million Malaysians have the 2 major risk factors. Controlling these risk factors can reduce the risk of having MI in the future. 6.4 million people have hypertension. From those who have hypertension, only half of them know that they are living with the disease. From those who are aware with having the disease, 90% of them are having treatment, but only 45% have controlled blood pressure. 8 million adults have raised total cholesterol level among them. Among in 4 are aware, 1 in 4 are unaware of having such disease. Only 80% of high cholesterol patients seeking for medication, but only 63% have their cholesterol level controlled. Mostly people aged 40 years and above are unaware of having high blood cholesterol condition. 3.8 million people aged 18 years old and above have diabetes. Among them, 8.9% do not know that they are having diabetes. Within the states in Malaysia, Negeri Sembilan, Perlis, Pahang record the highest prevalence of diabetes. Half of adults are overweight or obese, and half of adults are abdominal obesity. Abdominal obesity is the condition when waist circumference greater than 90 cm for men and 80 cm for women. 21.3% of nations are smokers. 21% of smokers smoke cigarette, while 5% smokers smoke e-cigarette. According to the research, One in two people claim that they are exposed to second-hand smoker at restaurant without air conditioning. 31% of population said that they are exposed to second-hand smoking at the home, 27% at workplace, and 9% at restaurant with air conditioning. The smoking trend in Malaysia, smoking cigarette was found to be the largest risk factors of premature death among developing countries. Approximately 3 million deaths were reported per year due to the smoking-related disease. In Malaysia, there are about 5 million smokers aged below 18 years old. The research also found that 1 in 10 Malaysians aged between 13 to 17 years old were smokers. More than 50% of secondary school male students were caught to be smokers. Other than that, Malaysia's lower income population tends to pose a higher rate of smokers compared to the upper income group. Below 40 households spend 2.3% of their monthly income on tobacco and alcoholic beverage, which is higher than T20. It is estimated around RM70 per month will be used to buy tobacco. Other than CVD, smoking can cause cancer, respiratory disease, and chronic obstructive pulmonary disease. In 2019, there are 21.3% of smokers in Malaysia. The trend of smokers in Malaysia is decreasing for the past years. Around 4.8 million people aged 15 years old and above in Malaysia are smokers. Most of smokers in Malaysia are living in rural area. In average, people smoke 12.4 cigarettes per day. Majority of male smokers smoke 15 to 24 and female smokers smoke 5 cigarettes per day. The monthly expenditure, as you can see from the graph, is decreasing from 2016 to 2019 for tobacco and alcoholic beverage, and you can see from the graph also the smoking trend in Malaysia is decreasing yearly, and this is maybe due to the government law that prohibited to smoke at the restaurant, and also percentage of deaths due to smoking is higher at every country, is higher than 10%. So how we want to prevent MI? By having control blood pressure, eat a healthy diet, maintain healthy weight, exercise regularly, stop smoking and reduce harmful use of alcohol, and reduce the probability of having non-communicable disease. Having a healthy lifestyle such as often consumption of fruit and vegetables, smoking cessation, and regular exercise can reduce the risk of MI. Physical activities reduce the risk of MI. Having 30 minutes per week of exercise is suggested to reduce the risk of MI. Quitting smoking is one of the ways to prevent having MI. It was found that around 40% of smokers tried to quit, but only 4% to 6% succeed in their struggle. So based on the conclusion is CVD is the main cause of death worldwide. Among all the diseases in CVD, MI and stroke are the most common cases appear in the society. There are many risk factors that contribute to the high number of deaths due to CVD. Hypertension, cholesterol level, and diabetes are the main risk factors of CVD in Malaysia, followed by BMI and smoking. Smoking is the main risk factors of premature death worldwide and one of the significant risk factors of MI, and it is controllable factors. So that by controlling the amount of smoking or smoking cessation can reduce the risk of having MI or the risk of death due to MI. Lastly, there are many ways to reduce the risk of CVD, such as quitting or reducing smoking, having a healthy diet with regular consumption of fruits and vegetables, and having physical activities such as sport, jogging, and so on. So the recommendation from this study, a further study on modeling the risk of myocardial infarction related to smoking and myocardial is needed to understand the real association of smoking with myocardial infarction, especially among lower income individuals in Malaysia. Acknowledgement, thank you to Ministry of Health and Education for sponsoring this research. So, thank you.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Adli Wahida binti Murad, and today I'm going to present my research entitled Identification of Antioxidants and Active Compounds in Cow's Milk. For my presentation, I will explain about the introduction, experimental methodology, results and discussion of the experiment, and the conclusion. For the introduction, cow's milk is a milk that were derived from most taurine species. Most compounds that are present in this milk is carboxylic acid with a long aliphatic chain. The benefits of antioxidants present in this milk are protects the cells in the body from the free radicals, which is the unstable molecules that are produced by the body. For the objectives, firstly, to extract antioxidant and active compounds from commercialized cow's milk using liquid-liquid extraction technique. Secondly, to investigate the fatty acid compounds of antioxidant existed in the cow's milk samples using TCMS. For the methodology, commercialized milks, which are milk sample A and milk sample B, are chosen for the identification and comparison of antioxidants in both milk. Acetic acetate and hexane are used as the solvents to extract the antioxidant compound in milk. The milk sample, which are milk sample A and milk sample B, were measured in measuring cylinder. 20 ml of each milk were poured into 4 conical flasks. 20 ml of ethyl acetate and 20 ml of hexane were then added separately into the 4 conical flasks. The conical flasks containing mixture of milk and solvent were sealed with parafilm to prohibit foreign substance from entering. All the conical flasks were labeled and left in the chiller for 24 hours. All the milk samples were extracted by liquid-liquid extraction method using separatory funnel. 2 layers were formed from this technique in which the white layer contained water molecules and remaining chemical compound of the milk, and the transparent layer contained extractable compounds of the milk samples. The procedures were replicated twice to optimize compounds extraction. The excess solvent were then removed by rotary evaporator, in which the bath temperature was set at 76 degrees Celsius for the ethyl acetate, while the milk sample with hexane was set at 68 degrees Celsius. The extractable compounds left in rotating bottle were collected and placed in sample bottles. The samples that are collected were injected into KoSiST chromatography machine. The KoSiST chromatography spectrum for each milk sample, which are milk sample A with ethyl acetate, milk sample A with hexane, milk sample B with ethyl acetate, and milk sample B with hexane, were obtained as illustrated in Figure 1, Figure 2, Figure 3, and Figure 4 respectively. The KoSiST chromatography spectrum for each milk sample, which are milk sample A with ethyl acetate, milk sample A with hexane, milk sample B with ethyl acetate, and milk sample B with hexane, were obtained as illustrated in For the conclusion, commercialized milks were studied in order to investigate the antioxidants from the composition of fatty acids in each milk. Several types of fatty acids were identified in milk samples such as oleic acid, stearic acid, capric acid, and decanoic acid and palmitic acid. This study provides The data from the analysis of GCMS spectra of commercialized milk sample. That's all from me. With that, thank you.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nurhanini Nahlul Muna bint Yusuf. I'm from KoSiST Genus Insan. So today I'm gonna present to you about my study on spectroscopy analysis of antimicrobial compound in Centella acetica extract. So the next slide is on my introduction. One of the most generally utilized therapeutic plants in Malaysia is Centella acetica. Around the world, Centella acetica is available and is utilized as a wellspring of food, drink, and medication. Centella acetica has been ordinarily utilized in people's medication for a long time to treat a wide assortment of infections. It has been highly regarded since prehistoric times for its use as a medicinal plant. Centella acetica likewise one of the therapeutic plants that show antimicrobial properties as it has potential in wound healing activities. So, this research focuses on identifying antimicrobial compound in Centella acetica using spectroscopy analysis. The identification of the antimicrobial compound using this spectroscopy analysis will determine the antimicrobial properties in Centella acetica. So, for the methodology, I use liquid-liquid extraction method to extract Centella acetica leaves. Next, I use attenuated total reflection Fourier transfer infrared ATR-FTIR for the spectroscopy analysis of Centella acetica extract. Next, I will determine the functional group of Centella acetica by the spectrum graph. Lastly, I will determine the antimicrobial compound by the presence of functional group in Centella acetica extract. So for the results, using this spectroscopy analysis using ATR-FTIR, the functional group of antimicrobial compounds found in Centella acetica leaves was determined. The functional group was identified through the spectrum and wavelength shown by ATR-FTIR from Centella acetica extract. And this is the spectrum graph that I obtained from the Centella acetica extract from ATR-FTIR. Next, it can be seen that this functional group existed in Centella acetica leaves. These functional groups are found in vanillin, cinnamaldehyde, eugenol, and safrole, which indicates the antimicrobial compounds. This table shows the functional group and compounds identified from the Centella acetica extract. Thus, antimicrobial compounds are found in the Centella acetica leaves, which shows that Centella acetica leaves has antimicrobial properties. The conclusion is the spectroscopy analysis determined the antimicrobial properties of Centella acetica using FTIR by interpreting FTIR spectra beginning at the high frequency and by the presence of functional groups. The result for this research is the presence of functional group in Centella acetica leaves are vanillin, cinnamaldehyde, eugenol, and safrole, which indicates as antimicrobial compound in Centella acetica. Therefore, the usage of Centella acetica leaves in society needs to be expanded so that its nutrients can be utilized to the maximum for the benefit of the people and improvement to the national national economy. So that's all from me. Thank you. Assalamualaikum warahmatullahi wabarakatuh. My name is Kairina binti Idris and I am from College Genius Insight, University Science Malaysia. So today I will presenting my research titled Analysis of Antimicrobial Compound in Ziziphus mauritana extract using Attenuated Total Reflection Fourier Transform Infrared, which is ATR FTIR. Moving to the introduction slide, it is the picture of Ziziphus mauritana leaves, and this leaf is generally known as bidara among Malaysian, and it lives both in tropical and subtropical regions. It grows in forests, thickets along riverbanks, hills, and slopes in its native habitats. Then, the leaves are normally about 2.5 to 3.2 cm long with a fine tooth at the edge and 1.8 to 3.8 cm wide. Then the foliage can be evergreen or deciduous, contingent upon the atmosphere. Next, what is Fourier transform infrared spectroscopy? Okay, this machine takes an infrared range of assimilation or discharge of a fluid, gas, or solid. Then it gives crucial data about spectra. It can gauge the power of spectra over a wide scope of frequencies all at once. Then the electromagnetic radiation lies in the region between 2.5 millimeter, which is 4,000 to 400 centimeter for biochemical examination. This technique is used to measure and calculate the intensity of infrared radiation as a function of frequency or wavelength. Then we do have antimicrobial agent in this research, which we focus on eugenol and vanillin. Okay, eugenol is known as contagious operator for the treatment of infection, and it is most grounded, hostile to bacterial action against S. gordoni, and for Pheromonas gingivalis was shown by this antimicrobial agent. Next, we have vanillin. It has antimicrobial properties, and at a lesser degree, it has shown strong antimicrobial effects, which at dosage between 500 and 1000 ppm. Also, it more effective against fungi and non-lactic gram-positives than gram-negative bacteria. Furthermore, we do study about antimicrobial compounds. In plants, bioactive mixes are normally created as optional metabolites, which terpenes, tannins, flavonoids, basic oil, alkaloids, lecithin, and polypeptides as the principal bunches with antifungal action. But in this research, we focused on tannins, polyphenols, and also alkaloids. Then, what is actually the problem that make this study is important? So, in certain studies, the antimicrobial compound are studied but did not well related with traditional medicine such as Ziziphus mauritana. Then this study's objective is to focus on analyzing the antimicrobial compound in Ziziphus mauritana extract from the antimicrobial agent found in this extract. Next, we continue with the methodology carried out to do this research. We do extraction process and also characterization process. In the extraction process, the Ziziphus mauritana leaves is grinded with pestle and mortar and is dried and is extracted using ethyl acetate as the solvent. And we do the liquid extraction. After that, the characterization process are done by using ATR FTIR. Thus, the next slide is showing the result of the ATR FTIR in form of a spectrum graph. So in this graph, there is a peak, like many peaks. Then, to conclude, in table 1, it shows the functional groups and compounds identified from sample, which the frequency we obtain from the peaks and then we search for the functional group, and then these functional groups contained by the Ziziphus mauritana leaves. To be discussed, the functional group of Ziziphus mauritana was determined from the frequency identified by FTIR, and then antimicrobial agents were chosen, which is the eugenol, and vanillin, and both of it have similarity with Ziziphus mauritana in terms of their functional group. Hence, Ziziphus mauritana is expected to contain both of antimicrobial agents of eugenol and vanillin. So, Ziziphus mauritana is proven to have antimicrobial activity. Also, the effectiveness of this plant is further discussed with stating the bioactive compound found in the plant to relate with the antimicrobial agent suggested. Lastly, for the conclusion, this plant, which is Ziziphus mauritana, do contain antimicrobial agents which are eugenol and vanillin. Thus, it contains antimicrobial compound which support the statement of this plant do has antimicrobial activity, and the value of this plant can be shown with strong evidence of scientific research, as we do carry out laboratory method to do this study. And the community can use it as one of the traditional medicine to inhibit microbes. Then it also can contribute to the country's economy, as it can be commercialized as traditional medicine with antimicrobial property. So that's all from me. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Hi and Assalamualaikum warahmatullahi wabarakatuh. My name is Shanatra binti Shahabuddin and I am from College Jenis Insan, University Sains Islam Malaysia. Today I will present to you my research entitled DNA-based method for Capra agaricus hircus of raw and factory-made milk. Let me start with my introduction. Goat's milk. Goat's milk actually contains more protein, carbohydrate, vitamins, and minerals compared to other milks such as cow's milk. It also contains nutrients that help to build and maintain bone strength, boost muscle growth, and support healthy weight. As our research is based on DNA extraction, our objective is to determine the authentication of commercialized goat's milk by comparing it with fresh goat's milk and using DNA extraction method with the Promega kit of genomic DNA. For our DNA extraction, the most common tissue used is actually blood, but for this study, we obtained the mammalian DNA from milk sample, which is simple and non-invasive. Non-invasive technique is used to extract the goat's DNA. Thus, this helped to reduce the stress in animals during collection of research material. For our Islamic perspective, Allah has actually mentioned about milk in the Holy Quran in Surah An-Nahl verse 66. وَإِنَّ لَكُمْ فِي الْأَنْعَامِ لَعِبْرَةً نسقيكم مما في بطونه من بين فرث ودم لبنا خالصا سائغا للشاربين. The word between excretion and the blood give meanings that different pathways inside the body of a mammal ensure all blood and feces and milk is not mixed. The blood flows through the veins, the milk goes to the udder, the urine goes to the bladder, and the feces goes to the anus. Each of them give different benefits but contain the same DNA. For our methodology, we used Wizard Genomic DNA Purification Kit, so we followed the instruction provided by the kit. Firstly, we added 3 microliter of RNA solution to the milk sample. We mixed it and incubated for 15 to 30 minutes at 37 Celsius. Next, we added 200 of microliter of protein precipitation solution, vortex and chill it on ice for 5 minutes. The sample is then centrifuged for 4 minutes. Next, for DNA precipitation and rehydration, we transfer the supernatant to a fresh tube containing 600 microliter of room temperature isopropanol. It is mixed gently by inversion, and the sample is centrifuged for 1 minute. The step is continued by removing the supernatant and added 600 of microliter of room temperature 70% ethanol. The sample is then centrifuged again for 1 minute. Next, the ethanol is aspirated and the pellet is air dried. For our last step, the DNA is rehydrated using 100 microliter of DNA rehydration solution for about 1 hour at 65 Celsius or overnight at 4 Celsius. We arrive at our result and discussion. We obtained that all DNA samples extracted from raw goat's milk were actually high purity and quality. Identification of species by mitochondrial DNA copies per cell because it is greater compared to nuclear DNA. The PCR amplification product shows that the bands were clear and single. No non-specific products or dimer fragments were found. Thus, the result of the amplification indicate that this method can be used to extract high quality of DNA. Thus, this is the result of our DNA extraction. On the right is our commercialized goat's milk and on the left is fresh farm milk. Lastly, for the conclusion, this study has shown that all of the milk samples from fresh farm milks and commercialized milks actually contains DNA, and the DNA extracted can be used for further studies in species determination to determine the different sequences of DNA in each milk samples. Thank you for listening. That's all from me. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. My name is Aisyah Akila and I am from College Jenis Insan University Science Islam Malaysia. So today, I would like to present about sequencing and annotated of goat's milk using bioinformatics tool, which is Cluster X. Goat's milk. What is goat's milk? Goat's milk is famous for its nutrients. They are rich in quality proteins, carbohydrates, fat, vitamins, and minerals. Goat's milk is lighter in cholesterol than any of the cow milk. DNA sequencing. DNA sequencing is the method of determining the order of nucleotides within a molecule of DNA. The main purpose of DNA sequencing is to identify and comprehending the inner structure of molecules in genes. Bioinformatics tool. Bioinformatics tool is the application of software, computing, and arithmetic in order to help resolving biological issues. it can use to conduct sequence or analysis of DNA structure in an organism. In this research, the bioinformatics tool of KoSiST will be applied to the DNA of commercialized goat milk and fresh farm goat milk from analyzing the data. Islamic perspective. In Surah Al-Mu'minun, verse 21, Allah has mentioned, A'udzu billahi minasyaitonir rajim. Wa inna lakum fil an'ami la'ibrah. Nuskikum mimma fi butuniha wa lakum fiha manafi'u kathiratun wa minha ta'kulun. And for you, in them, the drink are numerous benefits, and from them you eat. The milk mentioned is a fresh milk, which is 100% milk without any addition substance. From Tafsir Al-Muyassar. There are many benefits that we can get from the milk, which is fresh and pure. Materials and methodology. Collection sample. 14 milk samples have been collected from each species, which is Jamnapari and Sanen from every state in Malaysia. Commercialized milk from brand Nubian and brand UK Farm were collected from the north, south, east, west, and also Klang Valley of Malaysia. DNA sequencing. After the DNA of every milk samples were extracted and undergoing PCR, products were purified by InnoPrep Double Pure Kit, Analytic Jena, Germany. The samples then directly sequenced using applied biosystem 3730XL DNA analyzer. And then, the DNA sequencing were sent and done by Matrox STN BHD. Next, alignment of DNA. When the DNA sequencing for every samples were successfully done, the sequence of DNA were aligned using KoSiST version 2.1. First, download the KoSiST version 2.1 and then open the program. Next, open the file and choose Load Sequence. It is then followed by choosing the DNA sequence to be aligned. The format of the file must to be in text. After all, the data from the DNA sequencing have been put in the program. Open the alignment and select Do Complete Alignment to perform the progressive alignment of DNA.
So, this is the result of the alignments from the samples.
Discussion. As we can see, there is empty spaces between the sequence of DNA. This is because the DNA from the samples cannot be read by the technology used due to sequencing is a one-time binding event and the DNA strands that were taken to be sequenced contain very short strands. Also, the primer used is not a common primer. KoSiST has been chosen to be used because it uses an easy text menu system that is portable to more or less all computer systems. The result shows that majority of the DNA were as same as the control variable, which is Ovis aries species. It may differ a little because of different parents from the DNA. Conclusion. Every samples have almost the same composition of DNA but not totally the same. Based on result, majority of the commercialized goat meat samples contain more than 90% similar to the control variable, which is Ovis aries species. KoSiST is also proven to be the best bioinformatics tool for aligning the DNA sequence.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. I'm Dr. Amin bin Abdul Rahman from the Faculty of Medicine and Health Science USIM. Our topic is reintroduction of gut microbiota improves liver regeneration in germ-free mice. Our team include me, myself, Amin bin Abdul Rahman, Dr. Muhammad Hairul Hisham bin Ngatiman, Professor Anthony M. Wheatley, Nur Fatin Akila binti Rahman, Dr. Nur Fariha Manzur, Professor Fergus Shanahan, and Dr. Malini binti Muhammad. The gut microbiome is known to affect the pathophysiology of the liver via the gut-liver axis. Microbiota and microbial products have direct communication with the liver via the portal vein. Therefore, changes in the gut microbial composition has been implicated in various diseases such as that of non-alcoholic fatty liver diseases, alcoholic liver diseases, primary sclerosing cholangitis, and hepatocellular carcinoma. Furthermore, gut microbiota has also been shown to affect the regenerative capability of the liver. Mice with sterile gut, germ-free mice, and antibiotic treated mice demonstrate delayed liver regeneration following partial hepatectomy. Recent studies have demonstrated positive effects of reconstitution of gut microbiota in treating liver diseases. It has been shown that reconstitution of normal gut microbiota normalizes gut microbial dysbiosis associated with certain liver diseases and therefore leading to improved disease outcome. As recent studies have demonstrated positive effects of reconstitution of gut microbiota in treating various liver diseases, we would like to investigate the effect of reconstitution of gut microbiota on liver regeneration. We hypothesize that reconstitution of gut microbiota in sterile mice will improve liver regeneration following partial hepatectomy. Our objectives are first to investigate liver regeneration in germ-free mice that have been reconstituted with normal gut microbiota, therefore now known as ex-germ-free mice, and second to investigate whether reconstitution of normal gut microbiota improves liver regeneration by comparing liver regeneration between ex-germ-free mice, and wild-type control mice. 8 to 10 weeks old male mice of the C57BL strain were used. Mice were grouped into 3 groups: ex-germ-free mice, which were germ-free mice that have been reconstituted with normal gut microbiota, germ-free mice, and wild-type mice as controls. 3 to 4 animals were used per group. Reconstitution of the germ-free mice was achieved by cohabitation of the germ-free mice with normal wild-type mice. Since mice are coprophagic animals, transfer of gut microbiota occurs via the fecal-oral route, and after a few weeks, the germ-free mice have been reconstituted with normal gut microbiota. Mice underwent toothless partial hepatectomy under inhalational anesthesia, where the right medial, left middle, and left lateral lobes, which constitute 70% of the liver, or 2/3 of the total liver volume, were removed. Sham-operated mice, on the other hand, underwent gentle manipulation of the exposed liver lobes, which were then reintroduced back into the abdominal cavity. Sampling of the regenerating remnant liver were obtained at 0, 3, and 72 hours post-partial hepatectomy. The time point coincide with the different phases of liver regeneration. 3-hour time points coincide with the initiation phase of liver regeneration, while the 72-hour time point coincide with the proliferative phase of liver regeneration. Assessment of liver mass restoration was assessed at 72 hours post-partial hepatectomy, which coincide with the proliferative phase. Indices taken were liver weight to body weight ratio and normalized liver growth. Early regenerative response was assessed at 3 hours post-partial hepatectomy, which coincide with the initiation phase, and indices taken were protein concentration levels and gene expression levels of the cytokines and growth factors associated with early regenerative response of the liver. Protein concentration was assessed using Luminex protein assay method. Protein was extracted from the tissues using the TPER kit, and protein assay was conducted using the mouse premix multi-analyte kit. Reading of the microparticle beads was done by the Luminex MacBrix analyzer. Gene expression was assessed using the RNA sequencing method. RNA extraction from the tissue were done using the RNeasy mini kit. Library preparation was done using TrueSeq RNA sample preparation kit, and sequencing was done by the Illumina HiSeq 2500 system. Liver weight to body weight ratio of the germ-free mice at 0 and 72 hours post-partial hepatectomy was significantly lower compared to that of control wild-type mice, suggesting that germ-free mice were phenotypically smaller than that of the normal wild-type mice, while also suggesting reduced liver mass restoration in germ-free mice. Ex-germ-free mice, however, demonstrated similar liver weight to body weight ratio to that of control wild-type mice at both 0 and 72 hours, therefore suggesting that ex-germ-free mice were phenotypically similar to that of wild-type mice, while also suggesting normal liver mass restoration in ex-germ-free mice. Normalized liver growth by percentage was calculated by the liver weight to body weight ratio. At 72 hours post-partial hepatectomy, liver growth percentage of germ-free mice was significantly lower than that of wild-type controls, suggesting reduced liver growth in germ-free mice, while ex-germ-free mice demonstrated similar growth percentage to that of normal wild-type mice, therefore suggesting normal liver growth in ex-germ-free mice. Assessment of early cytokine, early protein concentration demonstrated that at 3 hours post-partial hepatectomy, germ-free mice had significantly lower concentration of interleukin-6 receptor compared to that of wild-type mice, and the concentration levels of all the proteins accessed was also lower in germ-free mice compared to that of wild-type mice, suggesting reduced regenerative response in germ-free mice, while ex-germ-free mice demonstrated no significant difference in protein concentration to that of wild-type mice, suggesting normal regenerative response in the ex-germ-free mice. Expression of genes at 3 hours post-partial hepatectomy demonstrated germ-free mice to have significantly lower expressions of HGF, interleukin-6R, and TNFR-SF1A genes compared to that of wild-type mice, therefore suggesting germ-free mice to have a reduced early regenerative response. No significant difference in gene expression was observed between wild-type and ex-germ-free mice. Clustering of the specimens also demonstrated that ex-germ-free mice had similar expression patterns to that of wild-type mice, while that of germ-free mice was significantly distant to that of wild-type and ex-germ-free mice. Therefore, we conclude that ex-germ-free mice demonstrated normal liver regeneration evident by normal liver mass restoration, liver growth percentage, and normal early regenerative response. Therefore, the introduction of gut microbiota in germ-free mice improves liver regeneration following partial hepatectomy, and subsequently confirming the role of gut microbiota in liver regeneration. This study was a collaborative study between the Faculty of Medicine and Health Science USIM and the Department of Physiology of the National University of Ireland, Galway. This study is supported by Fundamental Research Grant Scheme FRGS under the Ministry of Higher Education, Malaysia. Thank you.
Assalamualaikum and very good morning. My name is Muhammad Khairin bin Muhammad Hanafia. I am from Faculty of Dentistry, USIM. Today I would like to present my research regarding biosensors as point of care testing for diagnosis and monitoring of periodontitis. This is a comparative review. This is a table of content for my presentation. First is introduction, second routine diagnosis of periodontitis, third is application of saliva in periodontal diagnosis. The fourth is biomarkers of periodontitis, consists of bacteria-derived salivary biomarkers and host-derived salivary biomarkers. The last one is POC platform and mechanism, which is lab-on-chip platform and paper-based platform. First one is the introduction. Periodontitis, also known as gum disease, which is one of chronic inflammatory disease that triggered by bacterial infection. Periodontitis has significant impacts on oral function and quality of life. It is also associated with systemic disease such as diabetes and cardiovascular disease. The disease causes periodontal attachment loss, destruction of alveolar bone, and lead to tooth loss. The periodontitis occur in oral cavity involving tooth supporting structures, which includes gum, cementum, periodontal ligament, and alveolar bone. This is the prevalence in Malaysia. There are research between 10 years back, which is 1990 is 92.8% prevalence in Malaysia, but at the 2000 they are slightly decreased, which is 87.2%, but at the 2010 there is increased 94%, which is the highest prevalence in Malaysia. However, it is still challenging to diagnose and classify different forms of periodontitis cases due to old school methods to diagnose the disease and inability to predict the progression of the disease. Point number 2 is routine diagnosis of periodontitis. There are 4 main methods to diagnose periodontitis by the dentist. First is inspection. Inspection is to determine the severity of inflammation of periodontal soft tissue by examining color, shape, and depth of recession of the gingiva. The number 2 is palpation. The palpation is to analyze the progression of the disease by examining the stature of gingival and tooth mobility. And the third is probing. Probing is allow the dentist to measure the pocket depth, recession, bleeding on probing, and clinical attachment loss. The last one is radiography. Radiography is act as essential tools to diagnose the periodontitis. There are 2 types of radiography, periapical and intraoral radiograph. The third is application of saliva in periodontal diagnosis. The study has using a saliva sample, which is tool for diagnosis periodontitis because it is safe, non-invasive, and can be collected frequently. The number 2 is saliva contain promising biomarkers which correlate with clinical parameters of periodontitis. The third one is saliva able to combine with POC platform to be more accurate for diagnosis periodontitis. Also, it can be used outside clinical or dental settings such as in the rural areas, patient homes, and public areas. The fourth is biomarkers of periodontitis. There are 2 types of biomarkers, which is bacteria-derived salivary biomarkers and host-derived salivary biomarkers. For bacteria-derived salivary biomarkers, there are common bacteria that have been found in periodontitis such as Porphyromonas gingivalis, Prevotella intermedia, and Tanarella foresticia. The biomarkers has been developed from the bacteria by targeting their 16S rRNA. There are some other specific biomarkers that have been used to detect the presence of the bacteria, such as gingipins, which one of the proteases secreted by P. gingivalis, and the antimicrobial peptide as biological recognition element. The second one is host-derived salivary biomarkers. The biomarkers that have been developed from the destruction soft tissue, which release several enzymes and proteins involved in tissue degradation into the saliva. In this process, several cytokines that have been released, such as human neutrophil elastase, capsaicin G, autodendrogenic ameloblast associated protein, including proteases such as MMP8, MMP9, and salivary peroxidase. The biomarkers able to diagnose periodontitis and able to predict the progression and treatment of disease. This is the biomarkers that have been in the study. Number 5 is the POC platform and mechanism. POCT platform to better represent the current state of periodontal destruction, assess the risk of development and evaluate the recovery of periodontitis, point of care testing platform for periodontitis that have been established. There are several advantages of POCT platform, such as low cost, rapid detection, easy to use, able to conduct outside clinical or experimental area. There are two categories of POCT platform that have been introduced: lab-on-chip platform and paper-based platform. LOC platform is able to perform laboratory procedures at a small scale, involve multiple lab processes on a single chip with a minimum amount of reagent and high efficiency. Combination of LOC platform are to diagnose periodontitis, which focus on immunoassay and biomarkers. There are few examples of LOC platform to diagnose periodontitis. First, epidermolytic antimicrobial peptide-based sensor. Second, surface plasmon resonance. And the last one is surface acoustic wave technology, SAW. First, epidermolytic antimicrobial peptide-based sensor is to detect the presence of bacteria and with combination of miniaturized and integrated epidermolytic transducer and antimicrobial peptides. The second is the frequency range, which is in range 100 Hz to 1000 kHz with 100 millivolt amplitude voltage excitation. The detection limit has been detected, which is 10 to the power 1 colony forming units in KCI and 10 to the power of 2 CPU in artificial saliva. The advantage of this biosensor is the AMPs coupled with 3D IDA. Biosensors shown rapidly implementation, label-free and highly sensitive to detect the presence of bacteria. The second one is SPR. It's used to monitor antigen-antibody interaction, which is MMP9, that allow the sensor to evaluate changes of refractive index. The detection of the sensor will appear once analyzed molecule and specific ligand immobilized bound on sensor chip. The concentration of MMP9 range 10 to 200 ng per ml and the detection limit was 8 pg per ml. The SPR immunosensor is able label-free sensing system which promote practical procedure to measure and monitor MMP9 in biological samples. Another one is provide better understanding in interaction of molecular in life science, biotechnology, and medical science. The third one is an amperometric biosensor, which is biosensor coated with salivary peroxidase, in which detector assembled with a micropipette for injecting a sample liquid into flow detector and an electronic book. The sensor electronic book composed of an amplifier, a special designed peak detector, microprocessor, and display. Process involve sample injection, electrochemical detection, and data processing by pressing the plunger of the micropipette. The detection limit has been produced by this sensor is 0.5 ng per ml. The advantage by using this amperometric biosensor coupled with commercial available kits with peroxidase are able to introduce new portable electrochemical biodetectors for enzyme immunoassay. The fourth one is SAW. This biosensor coated with MMP8 antibodies to measure concentration in periodontal disease patient. The SAW is able to detect analyte in control box using analog signal. The biochip is composed input and output gold electrode linked by a gold film coated sensing area that has been built plain piezoelectric quartz crystal. The detection limit is 62.5 ng per ml with the effectiveness range 0 to 1000 ng mmp8 per mil. A real-time measurement able to modify protocols for chip preparation and allow to watch the reduction in time of analysis within 15 minutes. The last one is the paper-based platform, one of the cheapest technology with simple fabrication and independence from external instrument. There are, for example, of paper-based platform to diagnose periodontitis. First, magnetic nanoparticle biosensor, sandwich type lateral flow strip biosensor, and the last one is magnetic nanobit based assay. The first one, the magnetic nanoparticle biosensor, is coated with two types of salivary biomarkers, which is human neutrophil elastase and cathepsin G. is to measure paraelectric activity and magnetic nanoparticles using specific probes. The probes are coated with specific protectors that are will bound to magnetic bit from one end to end-gold sensor surface by the other end. When intact, the color of sensor was changed from gold to black. During proteolysis, the magnetic bits will cleave when attracted to an external magnet, which reveal the golden color sensor surface that can be observed from naked eye. This method able to reach sensitivity lowest 100 Fg per ml for captation G and 1 Pg per ml for human neutrophil elastase. The second one is sandwich type LFS biosensor is coupled with human odontogenic immobilized associated protein ODEM. Sensor will operate once the sample was added, then the sample will flow in the test device and pass through the conjugate pad into the nitrocellulose membrane and finally onto absorbent pad. The biosensor shows detection limit in buffer 8.32 nanomolar and saliva sample 14.59 nanomolar. The advantage is able to detect ODEM protein in the buffer but able to detect faster outcomes in human salivary. The last one is magnetic nanobit-based assay is labeled with gingipan specific peptide to diagnose periodontitis infection related to P. gingivalis. The sensor coupled with gingipan biomarkers and immobilized on a gold biosensing platform via gold thiol linkage, which resulting the color of gold layer change black. When the immobilized substrate cleaved by gingipans, the magnet will attract the magnetic nanobit peptide fragment so the golden surface color can be seen again. The sensor is highly sensitive and specific, which able to detect as low as 49 SFU of P. gingivalis within 30 seconds. The last one is conclusion. Based on the literature reviews, there are many types of biosensors that can be used along with specific biomarkers for the detection of periodontitis. Choosing a suitable biosensors and biomarkers depends on the purpose of research. On the other hand, there is a lot of biomarkers that have been developed for the diagnosis of periodontitis, and this combination able to make it fast, low cost, and easy to use for the detection. The biosensor also not only able to detect the periodontitis, but to predict the progression and treatment for the disease. That's all from me. Thank you.
I can help the older people to get back their normal brain aging. I can help the clinician to assess older people with MCI. I can help the policy makers to enhance the healthy quality among older people. I'm Nur Fazidah Mendes Muzi from College Genius Insight. Today, I would like to introduce to you blood pressure variability measurement during cognitive screening for older people based on literature review. Every 3 seconds, someone in the world develops dementia. It may be your grandma, your grandpa, or your parents. Due to the world aging population, Malaysia will be expected to have 590,000 older age who are aged 60 years and above suffer from the Alzheimer's disease in 2050. The medical health screening among older people usually was conducted every 6 months. So the cognitive assessment at the primary care becomes a crucial stage to detect the early stage of dementia, which is known as the mild cognitive impairment. Ministry of Health Malaysia 2009 reported that more than 60% of patients with the cognitive impairment receive their treatment at the moderate or severe stage of the illness. This, because of the development of pathology embraces the dementia symptom in many years, this is why these older people has late treatment. So during the primary care, cognitive assessment will start with the history taken from patient and the informant, followed by the physical examination. So this history taken is to ensure patient receive a thorough evaluation and helps build rapport. If there is an abnormality in the assessment, then clinician will perform the laboratory test, and if they are meet the criteria, they may refer to specialists at the memory clinic. During the cognitive history taken, modifiable risk factor of dementia will be investigated. So the clinician will ask few questions, including the medical background. For the example, do they have hypertension? Because hypertension is one of the uh, modifiable risk factor, and World Health Organization 2015 stated 1 in 5 men and 1 in 4 women are suffer from hypertension. Those who are suffer from this hypertension, they may increase 49% risk of dementia. So the hypertension usually measure one of measurement. This is the problem when it's come with the misdiagnosed or with the mass or white coat hypertension. Therefore, the need of blood pressure variability as the real blood pressure measurement. As we know, blood pressure is not constant variable. Rather, it shows marked spontaneous oscillation over short term, which is minute to day, or long term, which is month to year. So, over the past 2 decades, there has been increased attention on the blood pressure variability. Blood pressure variability is measured and classified through time range such as clinic visit, seasonal measurement, day to day, within 24 hours, and over seconds or minutes. So then all the BP data that was collected using the BP device will be calculated using mathematical concept. An increasing body of evidence has linked blood pressure variability to adverse cardiovascular outcome. The emerging evidence has now suggested the presence of relationship between increased blood pressure variability and the cognitive functions. However, the association between these two variables, which is BPV and the cognitive function, still in debating. This table shows the different type of blood pressure variability with the association to the cognitive function. We have a look at the long-term blood pressure variability. Long-term blood pressure variability refers to the blood pressure measured during visit to visit, season to season. Most of the studies produce negative negative association between standard deviation or coefficient of variance with the cognitive function, meaning that increased standard deviation or increased CV, which is coefficient variance, will decrease the cognitive function. At the clinic, BP monitors majorly employ automated spot check device and also followed by the mercury sphygmomanometer. The consensus discussion from the review study shows that long-term blood pressure variability is significantly associated with all cause and cardiovascular disease. Also, it will be increase the organ damage and also mortality. This is the major type of the blood pressure variability that was used to obtain the association between blood pressure variability and the cognitive function. Perhaps this is due to the assess method, maybe. Well, for the second types of blood pressure variability, which is midterm blood pressure variability, it is day-to-day measurement, and it was obtained using ambulatory blood pressure monitor in major study. It is one of the home blood pressure monitor, and it's attached to the belt around the wrist and connected to a cuff around upper the arm as the person move around through the normal daily life. So all the BP measurement will be collected and then will be measured usually using the standard deviation CV, coefficient of variance, and also average real variability. Right. So this similar to long-term blood pressure variability just now, the most study found that negative association between blood pressure variability and the cognitive function. Perhaps maybe it's due to the long-term period and also able to indicate the risk of dementia, which is the hypertension. Or for the short-term blood pressure variability, it is known as it is collected within a 24-hour period. And also similarly with the mid-term blood pressure variability, it will be collected, the blood pressure measurement will be collected using ambulatory blood pressure monitoring, ABPM. So the short-term blood pressure variability reflects normally being an indication of circadian variation. So over 24 hours BP, they will usually use standard deviation, coefficient of variance, and average real variability as the BP indices. And then the published study found blend association, which is there is a positive association, there is a negative association, and also there is no association between blood pressure variability and also the cognitive function. Meanwhile, for the last types of blood pressure variability, very short-term blood pressure variability, it is monitored due to the within beat-to-beat blood pressure variability. So it's measured using non-invasive finger cuff and blood pressure from heartbeat to heartbeat, and then due to the changes of the intrathoracic pressure such as respiration. BP monitors such as direct continuous intra-arterial recording coupled with the spectral analysis. Usually for the spectral analysis, they will transform into the MATLAB through the fast Fourier transform. So as a result, we can see that there is an image of the frequency indices, where this frequency and indices has been studied to investigate the influence of the autonomic nervous system. With that, as a conclusion, I am suggest that BPV is a potential non-invasive, low-cost and convenient predictor for the dementia. The needs of the standardization of the definition and methods of acquisition BPV for them to be used during the cognitive screening. So they are all my references. With that, thank you.